Hey everyone, I'm Dr. Marie McNeely, and I am pleased to welcome you to episode 621 of People Behind the Science. This week, we are rebroadcasting our interview with Dr. Rob Malenka, originally published on August 10th of 2015. Listeners, Rob is a trailblazer in the areas of synaptic transmission and plasticity in the brain. He is well known for identifying mechanically distinct forms of synaptic plasticity in the human brain, as well as for isolating a novel form of synaptic plasticity that may be involved in reward pathways. Rob also has investigated the role of dysfunctional neural circuits in diseases such as addiction and Alzheimer's disease. So listeners, get ready to learn more about Rob and his research in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am excited to be here today with Dr. Rob Malenka. So, Rob, thank you so much for joining us. How are you?
I'm good. It's a pleasure to be here.
Well, it is a pleasure to have you with us today. And I want to give our guests a little bit of information about you before we dive into our interview. So, listeners, Rob is the Pritzker Professor in Psychiatry and Behavioral Sciences at the Stanford University School of Medicine, as well as the Deputy Director of the Stanford Neurosciences Institute. He received his bachelor's degree from Harvard College and his MD and PhD in neuroscience from Stanford University School of Medicine. Rob then conducted postdoctoral research at the University of California, San Francisco, and he completed his residency in psychiatry at the Stanford University School of Medicine. Rob served on the faculty of the University of California, San Francisco for 10 years before he came to Stanford, where he is today. And now Rob is the recipient of the Medical Research Award in Neuropsychiatry from the Pasarow Foundation, as well as the Julius Axelrod Mentorship Award from the American College of Neuropsychopharmacology, and also the NARSAD Goldman-Rakic Prize for Outstanding Cognitive Neuroscience Research. In addition, Rob is a member of the National Academy of Sciences, a fellow of the American Academy of Arts and Sciences, a fellow of the American College of Neuropsychopharmacology, and also a member of the National Academy of Medicine. He has also served on the National Advisory Council on Drug Abuse and has also served as a counselor for the Society of Neuroscience. And now, listeners, Rob is here with us today to tell us about his experiences going through life and science. So, Rob, today we want to get to know you both as a scientist, but also as a person. So can you start off by telling us what you like to do with your time when you're not doing science?
I'm a reasonably serious tennis player, not as good as I would like to be, but when I'm not traveling, I try to play singles at least twice a week. Sometimes I skip out of work midweek and play, so sometimes even 3 times a week. I love living in the Bay Area. I've learned to appreciate really good wine. I don't drink as much as I would like, but I like to collect wine. I like to enjoy it with friends. I don't drink it by myself.
That's a good sign.
I love movies. I'm an escapist. I'm one of these people that cries on the plane when I watch romantic comedies, junky romantic comedies on the plane. I love science fiction. I love spy thrillers. I watch a lot of movies in my spare time. What else? In my youth, I used to backpack and hike a lot. I just haven't found the time to do that as much. And then finally, I love to travel. I used to do modest adventure travel, and now I still just love going and seeing new places, seeing the culture, interacting with the people, taking public transportation in various cities. And luckily, as a reasonably successful scientist, I get to travel a lot. I have friends all over the world through my scientific experiences.
Definitely. Well, wonderful to hear about all the things that keep you occupied when you're not working. And I must ask, what is the best movie as a movie buff that you have seen lately?
I really enjoyed this movie, Chef, which was about a professional chef who kind of gets fired from his fancy restaurant and then sets up a food truck. It wasn't a great film in terms of the cinematography or the style, but it was an upbeat, fun, life is good kind of movie. So that was fun.
Well, Rob, wonderful to hear about your life outside of science. And I like that you take to the tennis court as well to get out of the lab for a little while. So do you challenge your graduate students to tennis matches?
God, this is going to sound obnoxious. I haven't had any students or postdocs who are good enough yet. I play with a bunch of guys my age. When I was a postdoc, Way back at UCSF, I used to play tennis with the guy whose lab I worked with, Roger Nickel. On occasion, I'll play with a postdoc or a student and some other faculty colleagues, but I also have a group of non-science friends who I play tennis with.
Very cool. Well, great again to hear about your life outside of science, but I want to talk about your science next, Rob. Can you tell us how you describe your work to someone who's not familiar?
I tell them I'm a neuroscientist or a brain researcher, and I say part of my work studies how brain cells, which are called neurons, talk to one another. What makes the brain special is it's an information processing machine. And the cells in the brain basically are using a combination of electrical signaling and chemical signaling, are able to talk to each other through physical contacts that we call synapses. So one topic my lab studies is how do those synapses work at a really fundamental level, at a very molecular level? What are the proteins found at synapses and how do they behave? And then most importantly, my lab studies a topic that we term plasticity. So what makes the brain different than a computer is that the hardware in a computer, the wires and the circuits are hardwired. They don't change. Our brain circuitry, our brain wiring is changing all the time, and that's called plasticity. And it's changing in response to the experiences we have, to the substances we ingest, to the courses we are taught. And really, the plasticity of the brain is one of its most important and fundamental properties. And I'm most interested in how does that happen? How is it that when you listen to a boring interview on the radio, you might remember it for an hour or two. Mm-hmm. And that memory requires physical changes in your brain. Otherwise, you would not remember anything. So part of my work studies, really at a molecular level, how do those changes in the communication between nerve cells at synapses actually happen? What molecular mechanisms are underlying that? So that's part of my lab. And then the other part of my lab studies how does the nervous system use this plasticity and how does it get disrupted during various brain disorders like depression, like drug addiction, like Alzheimer's disease. So we study the synapses, the communication between nerve cells in specific brain regions that we believe that are strongly implicated in diseases, as I mentioned, such as depression or drug addiction or autism or Alzheimer's disease. And we try to understand how do those connections and what we call the circuits in the brain, how are they modified to cause some of the major symptoms of these kinds of disorders?
Well, very cool research, and I'm excited about diving into the details as we go through the interview. And perhaps I'm a little biased because I am a neuroscientist myself, but I think this stuff is so cool that you hardly need extra motivation, inspiration to keep yourself going to do the research that you do, to go through each day.
I completely agree with you because I can't imagine anybody who's not fascinated by the brain and how we think, why we feel the way we do, why we learn, all those big questions.
How it all works.
Yeah. How does it all work?
So do you, though, have a favorite motivational or inspirational quote to keep with you on your down days?
I have a cynical quote that I repeat all the time, which is a Henry Kissinger quote, which is academic politics are so intense because the stakes are so small.
Sounds about right.
Which is more a reflection of some of the irritations one has to put up as an academician, as a professor. But more positive inspirational quotes, I think it comes from Alcoholics Anonymous, and I can't quote it to you directly. It's about give me the strength to change the things in the world that I can change and the patience to not worry about the topics I can't change. So what I've learned over my now modestly long scientific career is that the things that really used to bother me as a young scientist, like getting harsh reviews or getting a paper rejected or getting a grant rejected or getting unfair criticism, and this just comes from experience, it rolls off my back much more than it used to. Because I know from experience that, in general, the system of science works. Good work will eventually get published and get acknowledged. Bad work will eventually disappear from the literature. And that while there's frustrations along the way, the system could still certainly be improved, but it basically works, and that's comforting.
Definitely. Well, I think these are great quotes to keep with you as you go through the ups and downs in science. And I'm curious to hear now about the people who've had a big impact on you or who've maybe inspired you over the course of your career. Do you have any role models you can point to?
Role models is maybe too strong a word, because I am a pretty experienced person. I've met a lot of well-known people, and I have to be honest, I don't really have any heroes or anybody I idolize other than Roger Federer, the tennis player.
Right. He's got skills.
He's got serious skills. But certainly, when I was an undergraduate at Harvard and I first started getting interested in neuroscience, I read a book from a famous psychiatrist and neuroscientist named Saul Snyder, who's still a professor at Johns Hopkins University. It was called Madness in the Brain. It was a layperson's description of the symptoms and causes of schizophrenia. That really got me interested in psychiatry and neuroscience. During college, I took a year off. I had grown up in Boston, went to Harvard, so it was my local school, and I had no idea what I wanted to do when I was an undergraduate. So after my sophomore year at Harvard, I decided to take a year off. For some reason, all my friends were from California. Many of them were from the Bay Area, so I decided to drive across the country with a friend and get an apartment in San Francisco and try to figure out what I wanted to do with my life. I was getting interested in what we now call neuroscience, even back then, and I was fortunate to be able to get a volunteer position in a sleep research laboratory at the San Francisco Veterans Administration Hospital with a psychiatrist named Erwin Bob Feinberg, who did clinical sleep research. And Bob was incredibly generous by letting me work in his lab and actually spending time with me. I was a 19, 20-year-old naive kid, but we used to just talk. He would spend hours a week talking to me about his research, about the brain and how drugs work in the brain, and it really had a major impact.
Oh, that's awesome.
So then I went back to Harvard and had a real focus on really more, back then, what was called physiological psychology, what we now call behavioral neuroscience. I started reading more. I started reading about psychiatry. I still didn't quite know what I wanted to do. I wasn't at all confident that I was smart enough to be a successful scientist. So I decided to go to med school to keep my options open. Came to Stanford because I had fallen in love with the San Francisco Bay Area on my year off, and this is a long time ago. I'm not going to tell your listeners how long ago.
It can be a secret. That's fine.
It can be a secret, a long time ago. And came to medical school, really didn't like medical school. So I actually did not enter Stanford in the MD PhD program. I entered in the MD program, but I came to Stanford because I knew they were a very research-oriented medical school, and I knew they encouraged medical students to do research. So after my first quarter of med school, I realized I'm not going to be able to get through med school if all I do is go to medical school classes. So I started looking for a lab to work in as somebody who had never done any sort of basic science research. And there I was really fortunate to find a position in a lab of a guy who back then was a very young assistant professor just setting up a lab, a guy named Jeff Kosis, who's now a professor at Yale. And he sort of worked closely with a slightly more senior but still very young neurologist by the name of Steve Waxman. And they let me into their lab, and I started working with Jeff, and it was just me and Jeff in the lab. We were doing what's called electrophysiology. We were making recordings from a variety of animal preparations, brain recordings, obviously. And it was a blast. It was just a lot of fun. Jeff made science fun, and it was so much better than sitting in medical school classes. And then I ended up staying in the lab, and I ended up getting a PhD with Jeff Kosis and Steve Waxman, both of whom, again, this is a long time ago, subsequently moved to Yale. Steve Waxman was the chair of neurology at Yale for many years. But Jeff and Steve had a major impact because it made me realize science could be fun. Doing experiments could be fun. It was a highly creative process. I didn't realize it at the time, but I now realize I had a talent for it. I actually wasn't bad at it. Mm-hmm. And then the last person I will mention before I stop this long-winded soliloquy.
This tribute to the people who've changed your life.
is the guy I did my postdoc with, who's a very famous neuroscientist, Roger Nichol. Some people call him Roger Nicole. Correct pronunciation is Roger Nichol. Mm-hmm. Roger is a synaptic neurobiologist at UCSF. And I decided to do my postdoc with him, and luckily he took me in. I learned a lot from Jeff Kostis and Steve Waxman as a PhD student, but what Roger taught me is how to really think of the big questions in neuroscience and how to attack the big questions with, I would like to believe, serious rigor and ferocity and passion, and not back off from the hard questions. So doing science with Roger, both as a postdoc and then we collaborated for several years when I became a faculty member, was a blast. And he really had a major influence. And then I'll stop there. There are a few others, but in terms of how I developed as a scientist, I think those are the key people.
Well, it's great to hear about some of these people who've had a big impact on you. And I love some of these ideas that you brought up of learning that science is a creative process. And I think that's something that a lot of people don't realize is there's a lot of room, especially when you're sort of in the hands-on science, really getting in there, asking these questions, trying to think of ways to solve them, ways to address them. I think it is way more creative than a lot of people realize.
Absolutely. I think especially people who are not in the sciences don't appreciate that the best science is often highly creative. I view my papers, my good papers, very much like an artist views his or her best pieces, best sculptures, best paintings. Some are so-so, but some I am really proud of because I created them in my head. I had hypotheses. I had thoughts about maybe it works this way. And if it works this way, how can I test that? How can I design an experiment? And then how do I craft these findings into a logical story that my readership, my colleagues are going to read and are going to really accept that the evidence I've accumulated really provides strong support for that hypothesis. When things are going well, it's a highly creative process. It's also putting together different methodologies or taking information from one field and applying it to another. So I'm not, unfortunately, a good musician or singer, but I really do believe it's a very creative process.
You're an excellent manuscript crafter. I like it. Well, Rob, wonderful to hear more about your journey and your process, kind of learning more about science as you've gone. But was there an earlier point, maybe before college, where you first had an inkling that science was maybe for you and this was the direction you wanted to go?
Yes and no. I mean, it will sound like absolutely yes when I tell you that my father was a physics professor in Boston at a place called Northeastern University.
Okay.
So I was exposed, and most of my parents' friends, not all, were physics professors in the Boston academic community, math professors, chemistry professors. So I was definitely exposed to an academic life growing up. And science, again, this is a long time ago, came easily to me. I was not interested in physics and math. I was always interested more in biology. But I was not seriously interested in it as a student. Although now that I think of it, what I just remembered is, sure, my mother sent me when I was 10 or 11 years old to demonstrations at the Boston Museum of Science. They had these things that 10-year-old kids could go to, and they would show you a snake and how it functions, or they'd tell you about the stars or something. Mm-hmm. But the truth is, when I was a kid, I did well in school because it kept my parents off my back.
Right.
But really, all I cared about was sports when I was a kid.
Interesting.
I had visions that maybe I could be a professional basketball player, or maybe once I started playing tennis, a professional tennis player. By the time I was in college, I quickly realized I did not have the aptitude or the talent to do that.
You were not the next Roger Federer, yeah.
And that I would have to figure out a different way to make a living. I wish I could say I was one of these kids, and I have friends who are like this, who when they were 7 years old were taking magnifying glasses and analyzing ant behavior or doing chemistry experiments in their parents' basement and blowing up things. That wasn't me. I just like to play basketball and soccer and baseball and tennis and goof off. I obviously was a decent student. I was able to go to Harvard. And as I said, when I got to Harvard, I was interested in sort of psychology, but I really had no idea what I wanted to do until I took my first course that was related to brain and behavior. And then bells and whistles did go off in my head. It was sort of like, wow, this is a fascinating topic.
Your aha moment there.
And then I was off. As soon as I discovered neuroscience, which when I was in high school didn't exist, I never had a course that ever talked about the brain. And it was sort of by accident I took it at Harvard. I mean, I was looking through the course catalog. All right, what am I going to take this semester? Mm-hmm. So that's the story.
Well, I think this is a great story for our listeners to hear who might be in the same boat. They didn't know when they were 7 years old that this is exactly what they want to do. They might be in college and still trying to decide that you can still become a very successful scientist, even if you haven't sort of been groomed for it from the get-go.
Absolutely. I think life is long and there are many twists and turns. I teach that to my students and postdocs, that just because you start on this path, you can take a left turn and then go back and take a right turn. You're not trapped. If you're clever and work hard, you can forge very interesting career paths.
Definitely. Well, Rob, now that we've talked about your history, your path to get to where you are today, I want to hear about one of these projects you have going on right now that you are just so excited about.
Oh, God, where to begin? I have a bunch of stuff. I'll tell you briefly about 3 different projects that are very different. One project, I'm very complimented that I get to work closely with a colleague here at Stanford whose lab is next to mine, whose office is next to mine, goes by the name of Tom Südhoff. Tom is quite famous because he won the Nobel Prize about 2 years ago. I would characterize Tom as being one of the world's leading, if not the world's leading, molecular synaptic neurobiologists. And what I mean by that is Tom won the Nobel Prize and perhaps even deserves a second one because he discovers proteins that have very important functions at these things we call synapses, at these contacts between nerve cells, which is where the communication between nerve cells happen. For many decades, I've been studying a phenomenon. The jargon we use is a major form of synaptic plasticity. As I mentioned earlier in the interview, the brain is plastic. It's changing all the time. And one way it's changing is that the communication between nerve cells, it can be modified. And we talk about the strength of that communication. So 2 nerve cells are talking to each other, and that communication can be really strong, meaning the volume sort of on the telephone line between the 2 nerve cells is really strong and very clear, or it can be very weak. And the strength of that communication can be bidirectionally modified. It can get bigger and louder, or it can get weaker and less loud. And we believe that this synaptic plasticity is at the root of brain plasticity. So I study a phenomenon in slices of the rodent brain by which different patterns of activity can strengthen that communication between nerve cells. And that phenomenon is called long-term potentiation, or LTP is the acronym. And it stands for long-term potentiation of synaptic transmission. And it turns out many people think this is key to learning and memory and key to all sorts of different forms of brain plasticity. So with Tom Südhoff's collaboration, we have a project ongoing where some of the proteins he discovered that he and the field thought only worked on one side of the synapse. The thought was these were only involved in the release of chemical messengers, things called neurotransmitters. But what we found is that these molecules are also very important on the other side of the synapse, that they're functioning in a very different way and in a very important way for this form of plasticity, synaptic plasticity, that my lab studies. So we're using very sophisticated molecular genetic manipulations of these proteins in mouse brains, and we're studying how they function in this form of plasticity called LTP. So that's one project I'm pretty excited about. And then the other project I'll tell you about is, as I mentioned earlier in the interview, another part of my lab looks at brain areas and circuits that we believe are pathophysiologically important for the symptoms of major illnesses like depression and addiction and autism. Some of these circuits and brain areas are part of the brain's so-called reward circuitry. And I guess for your audience, I have to explain that we now know that there are connections in your brain, areas in your brain, or what we call circuits in your brain, that are particularly important for telling us this feels good, this is rewarding, this is reinforcing. And these are evolutionarily primitive circuits. We believe they evolved to promote survival of various species. And it's kind of obvious that you need something in your brain that tells you food tastes good and is good when you're hungry. Water is really good and tastes good when you're thirsty. Warmth is good when you're cold. Sex is good most of the time.
Right.
And that doesn't happen magically, at least if you're a neuroscientist like myself. It's not magic. There's something in your brain telling you, whoops, that feels good. And it turns out there are specific areas in the brain that seem to be activated by highly reinforcing stimuli that presumably are telling us that these things feel good. And what's fascinating is that these circuits are plastic, just like the way I was talking about brain plasticity. And an obvious example of that I like to use is if you're an American, most of us celebrate Thanksgiving dinner. And if you think about Thanksgiving Day, you wake up in the morning and you smell the turkey cooking, you smell the pies baking. Mm-hmm. There's a lot of anticipatory, fun stuff you're thinking about. You're looking forward to eating that turkey and pie, hanging out with your friends and family. Many of us, like myself, starve ourselves so we could really enjoy it.
See, we do the opposite. We're like eating marathon all day, but
yeah, you start eating and it's highly rewarding.
Right.
As long as the turkey wasn't burnt and the pies weren't overcooked, it's delicious and you have a great time. It's an event that many of us in the United States really look forward to and have very fond memories of. So these stimuli were highly appetitive, meaning you really wanted that stuff, highly rewarding. But depending on when you eat, by 7 or 8 o'clock that evening, you are stuffed. And those very same stimuli, the smell of the turkey, the sight of the pies, rather than being rewarding, are at the best neutral and, in fact, can even be aversive. The last thing you want to do is eat more pie. The last thing you want to do is have another piece of turkey. Right. And I am fascinated by how does that happen? And it has to be that circuits in your brain are changing because it's the exact same sensory stimulus. So this is a long-winded way of saying I study the plasticity in this reward circuitry, and we recently have done work asking the question of, another thing that's highly rewarding and reinforcing for most people is hanging out with your friends.
Absolutely.
Most of us would rather go have dinner, go have a drink with family or friends. Most people, most of the time, don't want to go to that restaurant completely by themselves and sit in a corner. And again, it's kind of intuitively obvious why circuits in the brain evolved to make social interactions, most of the time, rewarding, reinforcing. And then the question is, how does that happen? Why are those social interactions reinforcing? And this is particularly important for certain brain disorders like autism spectrum disorders and certain features of diseases like schizophrenia, because as I'm sure you know, one of the cardinal features, for instance, of autism spectrum disorders is that people with autism don't interact normally with other people. And there's lots of reasons for that, but one possible reason is they don't experience the reward of social interactions. So this kind of thinking led my lab to do experiments where we started looking at how do social interactions in mice influence reward circuitry. And what we found is that this neuropeptide, I'm trying not to use jargon, that has been in the lay press a lot, called oxytocin. And what we found is that oxytocin can modulate the synapses and the circuits that are part of this reward circuitry. And we're now pursuing that topic. And we published a high-profile paper on that topic showing one way that oxytocin can modify a key component of this reward circuitry, which happens to go by the horrible name of the nucleus accumbens. Mm-hmm. But now we're following up on that work where we're doing lots more work using the modern tools of neuroscience to define where in the brain oxytocin is acting to enhance social interactions, to enhance social reward or social reinforcement. We're doing experiments where we can activate cells that release oxytocin, and we can genetically delete the receptors for oxytocin in specific brain areas, and it's a lot of fun. And then the last project that's related to this is we are actually beginning to study the neural basis of the actions of a drug which is called MDMA, which is also known as ecstasy. And as some of your listeners may know, a prominent psychological effects of MDMA or ecstasy is it enhances social interactions. It's an empathogenic substance. It actually really does increase a human being's ability to have empathy for another human being, and it really does enhance your desire to have a positive social interaction. So I've been fascinated for years by how does that actually work? And it turns out that MDMA, according to the scientific literature thus far, may actually influence some of the chemical messengers, including oxytocin and another one called serotonin, in the fashion that is related to the type of work I was talking to you about before. So we're actually giving mice MDMA, and we're trying to use the most modern tools of experimental neuroscience in mice to really elucidate the molecular, synaptic, and circuit actions of MDMA. So I will stop there. That's probably a lot more than you wanted to know.
Well, it's so great to hear more about your work, Rob. I think we could talk for hours just about how fascinating the brain is, this amazing process of neural plasticity, and all these big questions that it can really answer. So starting to get at some of these things that you're giving very great, relatable examples of, that scenario at Thanksgiving where you do not want to look at another morsel of food.
Yeah, it's how does that happen? And I really think these are fascinating and important questions.
Definitely. Well, great to hear more about your work. And I think we've talked about a lot of the exciting aspects of your career and about your journey to get to where you are today in science. But I want to hear about the dark days, the tough times, because I think these are so important to allow people to realize what the journey of a scientist looks like and to show that we also have failures. The successes are not the whole story in science. I think there's always so many failures, challenges, things that came before them. So can you share an example from your history?
As an experimentalist, especially when I did my own experiments as a PhD student, as a postdoc, and for the first 10 years I ran my lab, there were tons of times where experiments just weren't working, where I would try something and wouldn't work, or I couldn't get the methods I wanted to work. I will say again that I was very fortunate working with people that I mentioned earlier, in particular the people I did my PhD with, Jeff Kosis and Steve Waxman, and then the person I did my postdoc with Roger Nickel, of having wonderful role models. So during my PhD, it was just working with this guy, Jeff Kosis. Jeff was an eternal optimist. The glass is always half full type of person.
It helps to have those people around, I think.
It really did, because when I had weeks where things were not working, he was a cheerleader, saying, it's going to work. Don't worry about it. Let's try this. Let's try that. Just endlessly positive enthusiasm, which I really try to impart to my students and postdocs. Roger Nickel was sort of a different type of example where he wasn't eternally optimistic, but he had an incredible internal drive and passion to find the answers and wouldn't let anything get in his way or our way. So if this experiment didn't work, let's think of another one. We're not going to give up. We're never going to give up. We're going to find a way to solve this question. The other thing to appreciate about at least my journey is I was also fortunate, but this was by choice, to choose a primary methodology, at least back when I was in training and forging my career, which is this methodology of electrophysiology, where it's real-time data collection, where you're looking at the electrical activity or the synaptic activity within nerve cells in real time. And the reason I bring that up is for the type of work I was doing, especially as a graduate student, postdoc, and as a assistant associate professor, the failures were quick.
And you knew right away, yeah.
I knew right away or within a week or two that this wasn't going to work, so I could change something very quickly. I was not, and this was intentional, mostly because of my intellectual interest and my personality. I like doing those kinds of experiments that gave me immediate feedback. So I could figure out relatively quickly things weren't going to work and change things relatively quickly. So I never had to go through periods of months and months, a year where nothing worked.
Sure.
I've been also very fortunate that once I started my own lab, to have really great students and postdocs in general who often, when I wanted to give up, they weren't willing to give up.
Gotcha.
And they kept the pedal to the metal, as we said, and helped me get through the hard times. So, yes, there were dark times, but I have been very fortunate in my career that I've never had to go through a year, 2-year, 3-year period where nothing was working. And like I said, it was my role models and my own internal drive and passion that got me through those. I'm not going to give up. I'm going to try this or that. But I do teach my students and postdocs now that as a scientist, you have to have very thick skin, not only because you are going to face a lot of failures. For every successful experiment that gives you some great set of data, there may be 5 that don't work. So you need thick skin to just get through that, to have the passion and drive, and also thick skin to deal with all the criticism you're going to get.
Absolutely. So was there ever a project or an idea or a question that you really did just had to give up on?
Oh, yeah, of course. I have several examples that I gave up on, I think, as a young assistant professor in particular, because that is probably the most stressful time of an academician's scientific career. As a grad student, you're protected. You really don't quite know what's going on.
Right.
As a postdoc, you have pressure. You want to produce something, you want to succeed, but you're not trying to do everything yourself. You're not on your own. But at least in the United States system, as an assistant professor, it's wonderful that you now are on your own. You're running your own shop. You're running your own business. But engendered with that, as part of that, there is a lot of stress that you have to get something done. So yes, back in the day when I started my project, I spent a little while, I had somebody working with me playing around with viruses to try to manipulate proteins in nerve cells and in mouse brains. And using viruses now is a very standard methodology, which, among other methodologies, has really revolutionized our ability to manipulate the rodent brain. But I was trying to do this.
It was too soon.
In the very early days,
yeah.
because I saw the advantages, and I gave up on using viruses and those sorts of methodologies after only trying for maybe 4 or 6 months. And in retrospect, I wish I had hammered away at it for several years, because I really could have done really cool stuff before anybody else had done it. And I have a few other examples like that. But you live and you learn.
Yeah, definitely. So we don't just want to talk about the tough times in science. I know, like you said, these dark days, these challenges, these difficult times are often outweighing the successes in terms of sheer number. But I think the successes are what we live for as scientists, these great discoveries, these exciting moments. So can you share one of these moments of success? And this can be either a big success or even just a little victory along the way.
One example from slightly earlier in my career was, I was talking earlier about a recent project I'm excited about, and the study of this synaptic plasticity phenomena we call long-term potentiation, or LTP. So in the late 80s, early 90s, I was involved in a very big controversy about how this phenomenon actually worked. And I was arguing and almost battling some very smart, influential, and powerful neuroscientists who had a different opinion than I did about how this might work. And through reading the literature, and I won't go through the details, an idea was put in my head that, maybe this is how it works. And I was fortunate to have a very talented postdoc, and we started thinking about experiments to do to address this hypothesis, and it was called the silent synapse hypothesis. I was working independently at that point. I contacted my ex-postdoc mentor, Roger Nickel, and we hadn't worked together in 5 years. I had left his lab. I established independence. I was doing my own thing. He was doing his own thing. But I just knew he would be excited about these data and this kind of idea. And so we started doing experiments, and we were able to obtain evidence that this idea really was true, this so-called silent synapse hypothesis. And we published what I would say what is now somewhat a classic paper in this field. And that then led to this whole idea that these proteins we call glutamate receptors, which are recipients of the chemical messenger that certain nerve cells release, actually these receptors do not just sit at synapses, these physical contacts between nerve cells, but these receptors can actually move around. They can move into synapses, they can move away from synapses. And I will say that one finding in the follow-up work we did kind of established a whole new field in neurobiology, which then became a very major field and people are still pursuing. So that's one success from many years ago.
Well, I think that is a really exciting success. So how did you feel when you got that first result that indicated that you were really onto something?
It was a blast. I have been very fortunate that I've had several eureka moments. It's a rush. It activates your reward circuitry in a big way, especially when it's something you've been thinking about yourself, that you've created in your own mind, this hypothesis. Maybe it works this way. And trust me, at least in neurobiology, most of the time you say, maybe it works this way, and you're wrong. But every once in a while you say, maybe it works this way. And this would really explain a lot of findings of the field or I have not been able to put together in my own head. And then when one is involved in doing an experiment that says, hey, it really does work that way, that's exciting. Or I've also had experiences when I used to do experiments myself. And I will say there is nothing like doing an experiment oneself and seeing some results maybe for the first time, maybe before anybody else has ever seen that result. And appreciating that maybe that is telling you something really cool is going on. That's very exciting. That's the one aspect of doing experiments myself that I miss, the real-time interaction with the data. I still get excited when one of my postdocs or students brings in some new sets of data, and I look at it and go, wow, if that is really true, if that's a real result, that suggests this, this, and this. But it's not quite the same as when I used to do it with my own two hands. And that's what got me to be a scientist, is the fun of doing that myself.
Actually getting your hands on the data, actually producing the data yourself, I think is a huge part of the rush, right? Because you were there from the ground floor.
Absolutely. That's the one thing I regret about stopping doing experiments myself, that rush when things are working. I don't miss all the frustration when things aren't working well.
Right. Well, Rob, I think you did a great job, though, of capturing here the essence of the ups and downs that you experience as a scientist. And I think these are some of the major ones, you know, and I think these discoveries, these aha moments are just so exciting. And to be able to have had several of those within your career is phenomenal. So great to hear about those. And we've chatted a little bit about your life outside of science. And now I want to hear more, Rob, about what's on your bookshelf. I would love to get a book recommendation from you, something you think myself and our listeners should definitely check out.
It matters what kind of books you enjoy reading. I read a lot of magazines. I don't read as many books as I would like, although I go through periods, especially when I'm traveling a lot. I'll just read voraciously. So I just finished, I think this may have taken me 2 years.
Oh, wow.
It's a 3-volume set written by Robert Caro, biographies of Lyndon Baines Johnson, LBJ, our president. For your younger listeners, from 1963 to 1968. He was vice president with JFK, John Fitzgerald Kennedy. Kennedy was assassinated, and I'm old enough to vividly remember when JFK was assassinated. I was very, very young.
Right.
And these biographies, both because I remember the mid-'60s and the late '60s. I was very young, but I remember them. I remember LBJ, and these biographies are endlessly fascinating as a nonfiction story, both because they describe in exquisite detail the workings of Congress, because LBJ started out as a representative in Congress from Texas, and he became a senator from Texas, and then he became the vice president of the United States, and he became president when JFK was assassinated. So just the workings of Congress and politics at a federal level are fascinating. And then LBJ as a incredibly unusual and complicated personality was really fascinating, both just as one of our ex-presidents, but also me being a psychiatrist. It was not all of the books, because these are long, dense books, but a lot of them were like reading an exciting fiction novel. So that's one set, but you have to be dedicated to get through these books.
Right. It sounds like it's an investment. Yeah.
It's an investment.
Well, that is a great recommendation. And I like this idea of getting into politics, almost bringing the history to life. And I think politics for a lot of people in terms of the working of Congress, things like that are pretty black box for the average American.
That's why these are very informative. I really learned a lot about the political process and how important it is, and yet probably not surprisingly how convoluted and complicated it is.
Sure. Well, awesome recommendation for myself and our listeners. We'll put that out on our website for people to find there. And we've talked a little bit throughout our interview about travel opportunities. You mentioned like to get a little bit of reading in while you're traveling. And I know as a scientist, you get quite a few opportunities to see the world, to go to different places. So can you tell us about one of your favorite places that you visited for science?
I happen to have just come back from a trip 3 weeks ago where I had the fortune to participate in a very small, intimate meeting, maybe 20, 25 people, on the Greek island of Santorini. And I know Greece is in economic turmoil, but at least when I was there a few weeks ago, there was no indication of that whatsoever. And those of you who have been to Santorini know it is a spectacularly beautiful island. So the meeting was held at this hotel up high on what's called the caldera, which is the edge of an extinct volcano that juts out of the Mediterranean Sea 1,000 or 2,000 feet. So we were staying in hotels with panoramic views of the Mediterranean. We would have our discussions. It was a very discussion-oriented meeting, sitting on balconies overlooking the Mediterranean. It was just fun. It was fun socially, it was fun recreationally, and it was fun scientifically. And I could keep going. I've been very fortunate to be invited around the world to attend scientific meetings. So I've been to a lot of really fun places.
Well, this sounds like an incredible experience. And I think it's kind of nice to get outside of your normal surroundings to have these discussions, you know, get yourself out of your comfortable, familiar area and get yourself open to new ideas. So very, very cool.
I will say again to your younger listeners, what is fun about a scientific career, and I'm sure there are other careers in which this can happen, is science is an international avocation and passion. So as I think I mentioned earlier, I've been fortunate to be successful enough. So I have met scientists from Western Europe, from China, from Japan, even some from African countries, from South American countries, and it's an international language. So you can sit down with a scientist from France, from Italy, from Chile, from Japan, from China, and you immediately have a common language that you speak. It's a lot of fun. Definitely.
And that bond over something that you're both excited about, I think, too.
Exactly.
Well, fantastic to hear about some of your travel opportunities, Rob. And in addition to the travel opportunities, we do like to talk about the people, kind of showcase the human side of science, because every day you go into lab and you're working with people. I mean, science is done by people, and these people in your lab groups, you get a variety of personalities, but there's so many brilliant, creative, funny, and fun people in science today that I think it's hard not to get some quirky lab traditions that spring up, or even just hilarious memories you've shared together. So can you tell us about one of your own traditions or memories in science?
I try to run my lab with positive motivation. The best advice I ever got was from a professor of neurobiology here at Stanford who was on my PhD qualifying committee, who in his day was quite well respected and famous, a guy named Dennis Baylor, who worked on the biophysics of phototransduction, how it is that the photons that allow vision to happen, how they invade the retina and activate certain cells in the retina. And at some meeting we had, what he told me is the reason to do science is when all is said and done, it is fun, that you like it, that you have a passion for it. Because what he told me, and this was so true, is that you're going to have dark days. You're going to have days when nothing works. You're going to face criticism frequently, because that's what scientists do. We criticize each other. We look for the holes in each other's data, in each other's conclusions. And so what he told me is, well, in today's world, if you're entrepreneurial, maybe you'll get wealthy. You'll make a decent living, but you're not going to necessarily become rich from it. But the reason to do it is that most days you like coming to the lab. You like coming to work. You like thinking about the questions you're addressing. Mm-hmm. You like talking about it. You like the puzzle that it poses. So that was the best advice, that do science because when all is said and done, you like doing it and it's fun. And if you don't find it fun, there are easier ways to make a living.
Right.
So having said that, the people who have worked with me will tell you, I can be a pretty harsh critic. But I do that because when we try to publish our papers, we're going to get harsh criticism. So we might as well do it to ourselves first and try to find the holes in our own stories. But on the other hand, I also try to make it a fun experience. And part of the Malenka lab tradition is twice a year. We do outings, and we're very fortunate to live in the Bay Area. I've been a professor at UCSF or Stanford. So we have done raft trips where I take the whole lab and we go overnight to some river in Northern California. And as a community, we go rafting, wine tasting, where we do bike wine tasting. And we do this in a very safe way. We wear our helmets and we find little roads that we can bike on.
Sure.
So there are actually places in north of San Francisco where you can get on your bicycle and go from winery to winery and wine tasting. We're actually arranging now a camping trip to Big Sur in the next month or two.
Oh, wow. That's awesome.
We'll do that. So that's not a funny story, but what I've learned over the years is people in my lab really enjoy it, and it's a really good bonding experience. Whenever we do these outings, people come back to the lab with a renewed sense of friendliness and good energy.
Sure. Well, those are fantastic ways to get the lab together outside of the lab. And I think it is important to have that almost team building experience, that bonding, because you're working with each other so much during the day. It's easy for little things to spring up, people to get upset about things. But if you're having these fun experiences together, I think you're often willing to overlook some of the things that happen during the day. So
you hit the nail on the head. That's exactly correct.
Well, wonderful to hear more about your lab, Rob. And you mentioned the best advice you've ever gotten. And I think that is a fantastic piece of advice as well, to make sure you're having fun doing science. Which,
by the way, you have to sometimes remind yourself of.
Right. This is fun. Yeah.
Because science today, the year 2015, at least the science I'm involved in, is different than the science of 1980 or 83. Because, and you pointed this out earlier, science is done by human beings, and science is embedded in the society and the culture in which we live. And because of that, science is also influenced by how our society and culture changes. And people of my generation do appreciate that the world now is different than the world in the 1960s and 70s. The world is better in some ways, but it's also harsher in some ways. So science is a little bit more competitive now. Getting funding is a little bit harder. So I think because of that, it's even more important to remind you, as I say people all the time, it's better than a real job.
Right.
It gives you the opportunity to pursue passions and questions you're interested in. But one has to remind oneself frequently that you're doing this because most days it's fun.
Absolutely. And speaking of these passions and questions you're interested in, I know we've talked about some of the questions you've tackled over the years, but Rob, I want to know what the big question is for you. If we gave you everything, funding, staff, technology, feasibility, not a problem, what would you want to answer?
There are two big questions I want to solve. Both I alluded to when I was talking about the projects that are currently going on in my lab. One is, at a fairly detailed molecular level, I want to understand how this phenomenon of long-term potentiation, this major form of synaptic plasticity, how does it actually work in terms of sort of as a molecular machine? What are the key proteins? How do they fit together? And my model for this is some of the work that my colleague Tom Sudhoff has done, where he has helped define the detailed molecular mechanisms by which chemical messengers, these molecules called neurotransmitters, how they are released from a nerve cell in response to an electrical impulse. So that's one thing I would love to understand. And the other I alluded to too. If I had to pick one, it is how can we figure out some of the neural circuit basis of what I would define as social reward and, most importantly, empathy. And I think that is so important because, unfortunately, the human race, the human species, we have the ability to do astoundingly creative and productive things. We can build cities. We can create the internet. We can create phones that allow us to talk to almost any other individual in the world who also happens to have a cell phone. And that's remarkable. But simultaneously, as a species, we are capable of horrific behaviors. That's because, in my view, our brains evolved over millennia, and we have parts of our brain that tell us we need to be horrifically aggressive. We need to be horrifically nasty and mean. And simultaneously, we have parts of our brain that tell us not to sound like an ex-hippie, but we need to show love. We need to have enormous empathy for our fellow species. And I would really like to help figure out the brain mechanisms that would allow us to promote the good stuff in our brain and minimize the bad stuff. And I think by understanding the neural circuit basis of social reinforcement and empathy, we can think of ways of enhancing that good stuff, those feelings of positive experience that we have when we interact with other human beings, and we can start helping to diminish these nasty, aggressive feelings that we sometimes have. And I passionately believe that we need to do this for the survival of our species and the survival of our planet. And I think one way of doing that is modern neuroscience research, used in the best way possible. And eventually, when we really understand these kinds of brain mechanisms, there are going to be enormous ethical issues around how do we use that understanding in the most positive, best way possible.
Definitely.
How's that for an answer?
That is a great answer. I think these are some huge questions that you've brought up with. You can't even start to think about even the implications that they would have in society, the ethics, the policy, all these different things surrounding it. So very, very cool.
That's why I think neuroscience is where it's at.
Definitely.
I think we really need to support it and do more of it.
Well, great questions for myself and our listeners to ponder, Rob. And now you mentioned your favorite piece of advice, the best piece of advice you'd ever received. And now I want you to pay it forward and give us a piece of advice, or at least an inspirational note to end our interview on.
Find your passion and pursue it. Pursue it with creativity, pursue it with hard work, pursue it with intelligence, and hopefully it's a passion that is rewarding to yourself and societally productive. And if science is one of your passions, I would say pursue that in the same way. It's hard because there are so many fascinating scientific questions to pursue. I think the only negative is you can't do it all if you're an individual. So you can't solve world hunger and solve a malaria epidemic and figure out how the brain mediates empathy. We're all just single individuals. Unfortunately, you have to make some choices about what is that most fascinating question. Again, because I'm not a young man anymore, I don't mean to be trite, but remember that life is a process. That life is going to have its ups and downs, and you don't want to give up. You just want to remember that the good times are worth it.
Absolutely. Well said, Rob. Can you tell our listeners now where they can go if they want to learn more about you and your work?
The best place is the Stanford University website, which is just www.stanford.edu, and search me on the website. I have a web page through Stanford. And of course, if you just Google my name, a lot of stuff will come up. My papers will come up, pictures will come up, my website will come up. It's the miracles of the internet.
That's right.
Which didn't exist when I was growing up, but it is really a remarkable accomplishment. Definitely.
Well, listeners, check out his lab, what they're up to. And Rob, thank you so much for joining us today and sharing a piece of your story.
Oh, it's my pleasure. A lot of fun.
Well, it was a delight to chat with you today. And listeners, I hope you enjoyed our conversation as well. Thank you for joining us, and we'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.